Hello, hello. Hey, people, we're almost ready to rock and roll here. Let's see your faces. Come on, everybody. Bring them up. Bring them up. Bring them up, Kraylin. To Jovany. Reagan. Who is Reagan? Reagan. Have we met Reagan? I don't think so. Yeah, you're muted.
Yeah, sorry.
Okay. Oh, to Jovany. Are you dating Robert Russell? Because you're both kind of like this.
Oh, sorry, my computer screen.
No, you don't have to be sorry. That was a twofer. I got to tease both you and Robin.
Two for one.
In the business of cell biology, we call that a twofer. Thaisa, we haven't seen you in forever. Are you okay? Bring it up, Thaisa. Come on. McEnany is shy. He won't come up no matter what I say. Anthony Estevez. Anthony. Mina Fakir. Oh, Mina Fakir is here. That rhymes. There's Anthony. Good to see you, bud. Sophia Martin. Come on now. Sophia. You know. No, nobody gets to do anything until your face comes up. And everyone's going to blame you. Ah, there she is. And Barbara showed up today, which is always fun. Okay. Okay, so enough of that. Let's go ahead and do our quiz today. And again, when you open this, be careful. The ink might still be wet. I just finished this moments ago. And it's in Moodle. I'm going to bring it up here and then shut myself off. Oops, not that. Evo Devo. Okay, there it is. 3:30, that's today. And I will be back at 1:15. Tell you what you did wrong. Okay, let's look at the answers here. I just feel like a game show host when I do this. Group overrides. What am I doing? What am I doing? And a quiz. I think this one was pretty easy. At least I got most of the answers correct. Transcription factor phosphorylated by the RTK mechanism. Why, that would be our friend June. Small monomeric G protein, only one in the list, and it's RAS. Oops, wait. I did it again. Come on. I always do this. Okay, now you can see it. Transcription factor, June, G protein, RAS, mutant being by eliminating the kinase domain. Why, that would be a dominant negative. Kinase that MEK phosphorylates, MAPK. Kinase that phosphorylates MEK, that would be ref. A GEF. What would a GEF be? Oh, last but not least, a GEF. So that's that one. Kind of the RTK mechanism, but I looked at it a little differently and make it a little bit more interesting. Okay, oh, this was so easy. I hope nobody got any of these wrong. TGF beta mechanism, the hormone binds to the type 1, which then joins the type 2, which attracts the R-SMAD and phosphorylates it, which then phosphorylates the C-SMAD. That's pretty easy. And last but not least, in the nitric oxide mechanism. Now this one's a little bit, took a little thinking. In the nitric oxide mechanism, a hormone activates the phospholipase C. Is that correct? I believe so. Let's see, that IP3 produced caused, IP3 produced causes the release of calcium from the smoothie R, which binds to calmodulin, which activates nitric oxide synthase, guanyl cyclase, and arginine. Arginine, a pirate's favorite amino acid, arginine. Okay, so the nitric oxide mechanism, a hormone activates phospholipase C. The IP3 produced causes the release of calcium from the smoothie R. This binds to the calmodulin, which activates nitric oxide synthase, and this produces nitric oxide from arginine, which diffuses into other cells and activates guanyl cyclase. Bum, 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 bum. Hopefully you got all those right. They weren't very hard, in my opinion. Does my opinion matter? I believe so. Okay, let's start the learning up again. Okay, so today, what the hell are we going to talk about today? Okay, we're going to start out talking about the JAK-STAT mechanism, which is, again, pretty simple. I think it's neat. It's kind of cool. It is important. This is a mechanism used by cytokines, which are local modulators, local hormones being released by white blood cells. I believe the interleukins are cytokines. I'm not wrong. Okay, so JAK-STAT mechanism. So it's pretty similar to the tyrosine kinase receptor mechanism. Again, there's a lot of themes that are similar here. One difference is the receptors don't have a kinase domain to them. When the hormone shows up, the hormones clump together and that attracts kinases. One's called TYK2, tyrosine kinase 2. The other is JAK, which means Janus kinase. Not really sure why they called it that. I don't remember now. I used to know, don't know anymore. A lot of shit I don't know anymore, used to know. Okay, so again, like the tyrosine kinase receptor mechanism, they transphosphorylate. And oftentimes this phosphorylation not overactivates, increases the activation of the kinase domain. If you take my biology of cancer class next fall, I'll be talking about that, how the phosphorylation activates the kinase even greater. And then you get phosphorylation on other tyrosines in the receptor. So you can see the first phosphorylations go on the kinases themselves, the kinase activity increases, and then you get phosphorylation of the receptor. So again, similar to RTK, these are SH2 domains. So these SH2 domains then will attract the STATs, STAT1 and STAT2. These are an interesting molecule in that they've got an SH2 domain and an SH2 binding domain. So this Y here can get phosphorylated and become an SH2 domain. Here's your binding domain here. So this then comes up and binds to the receptor using its SH2 binding domain, and then the receptor phosphorylates it. So STAT1 and STAT2 get phosphorylated. Now you've got two proteins that have both an SH2 domain and an SH2 binding domain. They dimerize using those domains, and I kind of think that's neat. In my world, that's neat. And then that becomes a transcription factor. So it translocates into the nucleus and then turns on certain genes by binding to their upstream control elements. Okay, so pretty easy mechanism, I think. I've got some animations there on it, or at least one. Receptors, the hormone binds receptors, they dimerize. That brings the enzymes up, the kinases up, they activate each other, then they phosphorylate the receptor, and that brings the STATs up. The STATs get phosphorylated, they dimerize, and then it's a transcription factor. Pretty simple. Probably as easy as the TGF-beta receptor mechanism, not too many steps. Okay, now, if you don't mind, let's do something more complicated. The PI3 kinase mechanism. Excuse me. So we've talked about phosphotyloinositol, the membrane phospholipid, quite a few times already in this class. You can see how important it is in signal transduction in the cell. So just to review, because you will see this on the final, in chapter 13, or was it 14? 13, I talked about PI being phosphorylated to PIP2, and then that was used to bring CAB-Z off the microfilaments and blah, blah, blah. And then earlier in this chapter, we saw PIP2 being broken down to release IP3 and diacylglycerol, both of those being second messengers. Here's another role for PI3 kinase in, PI for phosphotyloinositol in signal transduction. So here is our, let me just explode this. Here's our phosphotyloinositol PIP2. And so here you've got your, so here's your diacylglycerol. And then you've got a phosphate linkage to this inositol, and it's phosphorylated on the 4,5 position. That makes it PIP2. And then this enzyme PI3 kinase comes in, phosphorylates it on the 3 position. So now you've got 3 big phosphates sticking into the cytoplasm. This is still in the membrane, this is sticking in the cytoplasm. So you can see this is like, holy cow, 3 negative charges altogether on phosphates sticking into the membrane there. You got to bet that's going to attract some attention. So there are a whole bunch of enzymes that have something called the plextrin homology domain on them. And it's called that because the first protein that was found with one of these domains was plextrin. And then they find out, oh, look, that domain is found on all these other proteins too. So they gave it a generic name, plextrin homology domain. Kind of similar to SH2 domain, which is the Sark homology domain 2. It's got 3 domains. This is the second one. It's found on a lot of different things, so it's given this general name to make it easier to grab. One big difference is plextrin homology domain only binds to this, right, to PIP3. The SH2 domains are looking for those phosphorylated tyrosines, but also some other amino acids that are going to make it more specific. So you can have different types of SH2 domains that will attract different proteins. This plextrin homology domain, that's all there is. There's just the one. Okay, so it's going to recruit proteins with this domain to the membrane. Here is your plextrin homology domain. The yellow thing is the protein part of the PH domain. And you can see the The protein has got K, R, K, K, R, K. So it's 1, 2, 3, 4, 5, 6. 6 lysines and arginines. These are positively charged amino acids. So these obviously are going to be attracted to PIP3, which has got 3 negative charges there, 1 for each of the phosphates. So that's how this plextrin homology domain comes up to the membrane and binds to that because of that interaction between the positives and negatives. And as I said, there are a lot of different proteins that will have this. Some GEFs, guanine nucleotide exchange factors will have it. Phospholipase C gamma has it. So the phospholipase C we saw that was activated by the G protein GQ is phospholipase C beta. This is phospholipase C gamma. It's going to be activated by the RTK mechanism. And so when you get that PIP3, you can activate phospholipase C gamma, producing IP3 and diacylglycerol. PDK1, PDK2, we'll talk about these shortly. It's phosphate-dependent kinase 1, phosphate-dependent kinase 2. They have PDK or pH things. And then you've got AKT, which is kind of a central enzyme in a lot of different processes. We'll see it here associated with this particular pathway. Won't talk about these two right now. Just put a whole bunch out here so you can see there are a lot of different things that are associated with this. This is the main thing we're going to talk about being involved in the PI3 kinase mechanism. And you can see PDK1 will phosphorylate AKT, and that'll be part of our mechanism as well. Okay. What else? And remember, whenever you've got a gas pedal, you've got a brake. So our brake in this mechanism is PTEN. PI3 kinase will phosphorylate phosphatidylinositol 4,5 bisphosphate into phosphatidylinositol 3,4,5 bisphosphate. That binds the pH domain. P10 will take that number 3 phosphate off and make it back into PIP2. So this is your reversal enzyme for this mechanism. It turns PI3 into PIP3 into PIP2. Okay. South Park. That's where I got that from. South Park. I was just talking about this yesterday. I picked up that in a South Park episode. The teacher. Okay, you're a dirty little monkey. Okay, never mind. Okay, so here is the PI3 kinase mechanism. So several different ways to activate PI3 kinase. In this particular case, we're going to talk about an RTK mechanism. Okay, so this is similar to what we saw with RAS. RAF, MEK, and all that. You've got an enzyme, a receptor like the epithelial growth factor receptor, nerve growth factor, something growth factor, PDGF, blah, blah, blah. That's your enzyme. Hormone comes along, binds to it, receptors clump together, transphosphorylate. One of these SH2 domains is going to be specific for PI3 kinase. So it's got an SH2 binding domain on it. It gets recruited to the membrane. Now, since it's close to the membrane, it's going to come in contact with PIP2. If you remember again from last week, PIP2 is, I'm going to make a number up, but let's say it's like 1 to 2% of the membrane phospholipid. So when it comes into one of these PIP2s, it converts it into PIP3. PIP3 then activates will bring up anything with a pH domain. PDK1, PDK2, and AKT all have pH domains. PDK2 can also be called mTOR, small m, capital TOR, mTOR. So that's another important signaling enzyme. I don't get into it too much. If you ever take Dr. Parker for a class, you'll hear about it a lot because he's into mTOR. He's an mTOR kind of guy. So anyway, PDK1 and PDK2 both phosphorylate AKT, and then it becomes fully active and can come away from the membrane. Now, there's this whole other part of the signaling pathway that we didn't talk about, and that's GSK3 beta, glycogen synthase kinase 3 beta. GSK3 beta, K kinase, it's a kinase, it phosphorylates a protein called beta-catenin. So beta-catenin, some people say beta-catenin, but they're wrong. I don't know how to pronounce it, I'm just saying that. Beta-catenin is a transcription factor, and it just has to be present to be active. It doesn't need to be phosphorylated or anything. Just as long as it's there, it's going to go into the nucleus and activate gene transcription. But it's kept low on a constitutive basis by the action of GSK3 beta. So GSK3 beta is constantly phosphorylating beta-catenin. Beta-catenin then gets ubiquitinated and degraded. So we've talked about that mechanism before in like IRAD and things like that. We'll get in detail in chapter 24. For now, just keep in mind that this is constantly being broken down. And then AKT comes along and phosphorylates GSK3 beta and inhibits it. So once you shut off GSK3 beta, beta-catenin increases in the cell really quickly, goes into the nucleus, and it binds to another protein called TCF-LF. And TCF-LF and beta-catenin become a transcription factor, and it turns on a bunch of genes. This is a really important mechanism in cell division control. That beta-catenin, TCF-LF will activate things like cyclins and CDKs that are important for controlling cell division, and we'll talk about that in chapter 24. So anyway, this whole thing here is called the destruction complex, and that's because this is here just to keep destroying beta-catenin. It also constantly inhibits GSK glycogen synthase, GS glycogen synthase, and we'll see that in the insulin mechanism. Most of this is going to be repeated when we hit the insulin mechanism next. So GSK inhibits glycogen synthase, and that's kind of where the name came from. But this is a different and probably more important role for GSK3 beta, and that's keeping beta-catenin low. But you've got these other proteins, WTX, axin, and APC, and these are all kind of holding the structure of GSK3 beta in place to keep it active. If you take any of these proteins off, then you lose control of GSK3 beta. It just stops working. And APC is actually named for a colon cancer. Adenomatous polyposis coli. So it's mutated out in people who have really bad intestinal cancers, large intestinal cancers, colon cancer. So that's what APC is. Axin's used in some other mechanisms as well. I don't really want to get into it. WTX, I don't know. It's just another protein there. They're all kind of like scaffolding GSK3 beta, holding it in the right conformation so it can act. And we'll see that's really important because the last mechanism we'll talk about today is the Wnt mechanism, and the Wnt mechanism also involves this destruction complex. So it's going to come up again later today. So let me just go through the whole mechanism again, and hopefully it will make sense. When there's no hormone around and the receptors aren't active, PI3 kinase is inactive. So this destruction complex is active. So it keeps phosphorylating beta-catenin. Beta-catenin keeps getting broken down, okay? So you've got that cycle of destruction and nothing happening, basically. Then the hormone comes around, binds the receptor, receptors clump together, transphosphorylate, produces an SH2 domain that will bring PI3 kinase up. PI3 kinase then converts PIP2 into PIP3, which then attracts PDK1 and PDK2 and AKT to the membrane because each of these has a pH domain. These then, the PDK1 and 2 phosphorylate AKT, which becomes active, and it comes down and phosphorylates GSK3 beta, which becomes inactive, and that allows beta-catenin to build up in the cell. And then it goes into the nucleus and turns on signal transduction. Okay. Any questions about that? Any comments? Would you like to watch a video on that? We still have a lot of time to kill here. Be nice to get a little feedback from you people. I don't know. I never know whether you guys are understanding what I'm saying or not until after I give an exam and oh, look at that. No one knew what the hell I was talking about. Isn't that interesting? Too late then. Oh, another thing, this AKT is also called protein kinase B. So at this point, we've seen protein kinase A, B, and C. And so you may expect to see them again on an exam sometime in your future. When is our exam anyway? Anyone know? I'm supposed to know, right? Am I supposed to?
Syllabus says April 16th.
April 16th. April 16th. 16th.
Last time I looked.
Is that what it said? Because that's a Friday, so that could be messed up.
I think the syllabus says the 13th.
Hang on a second. It says the 13th. That makes sense. Oh yeah, there it is on my schedule. April 13th. So let's see, next week, let's see, we've got this Thursday and then next Tuesday. So Thursday and Tuesday, we will do Xenopus early development and signal transduction involved in that. And then I guess the next Thursday, we'll do a review and then exam on the following Tuesday. That makes sense, doesn't it? I believe so. No one's answering me. That's okay, though. I'm not taking it personally. Okay, so that's protein kinase B or AKT. Just looking at my notes here, making sure I didn't forget anything. Okay, so the next mechanism then is the insulin receptor mechanism. And I've got the figure I have here is from your book. I'm not crazy about it. I made an animation that really rocks, but unfortunately, as I've told you before, all my old animations don't work anymore because they were made in Flash. So I can still show it to you, but I have to share it to you from my screen. So here's the insulin receptor mechanism. So again, this is very similar to the last mechanism. Big difference is like the output at the end and the receptor part at the beginning. Everything in the middle is pretty much the same. So the insulin receptor is not a single protein, it's actually two proteins, an alpha subunit and a beta subunit. So the alpha's got the hormone binding domain, and the beta then is got the transmembrane domain and the kinase domain. And tyrosines in SH2 domains in the tail here. PI3 kinase is going to be part of it. Here's another new one, IRS1, insulin receptor substrate 1. So that makes sense. You know, it's the protein that insulin receptor is going to phosphorylate, IRS1. So here's how this works. Insulin's around, so your blood, think about it this way. Say your blood glucose is normal, okay? Blood glucose is normal, there's no insulin around. So what happens is you've got an active glycogen synthase kinase in the cell. Oh, Jesus. Oh, God. Got active glycogen synthase kinase in the cell. It's phosphorylating glycogen synthase and inactivating it. So glycogen synthase is off, okay? Then your glucose level goes up, okay? So you eat a big meal, glucose goes up in your body, your pancreas releases insulin. Insulin binds to the alpha subunit, which then binds to the alpha beta. All this becomes active then. The tyrosine kinase produces phosphorylation sites here, attracts IRS1. IRS1 gets phosphorylated, and now you've got an SH2 domain that attracts PI3 kinase. PI3 kinase then phosphorylates PIP2 into PIP3. PIP3 then activates PDK1, PDK2, and AKT, PDK1, PDK2, phosphorylate AKT, it becomes active. So all of that part is the same as the last mechanism. Now here's what's different. If you remember when we talked about receptor regulation, I said there's always like this subcellular pool of vesicles that where things can go into the membrane, out of the membrane quickly from this pool of vesicles. Subcellularly, you've got a pool of vesicles that have got a glucose transporter in it, GLUT4. So this is just a passive glucose transporter like we talked about in chapter 8. This can go in either direction, either transport in, glucose in or out. So they are in vesicles here in the membrane. So when AKT near the membrane, sorry, when AKT gets activated, it phosphorylates a protein on that vesicle. The protein is called I don't have it here. I thought I did. Okay, so it phosphorylates a protein on the vesicle. That causes the vesicle to fuse with the membrane and put new glucose transporters into the membrane. Now glucose starts coming into the cell faster. So that helps to get the glucose level in the blood down. So that's what insulin's doing. Glucose goes up, it activates putting more glucose transporters in the cell membrane. That brings glucose into the cell, especially the liver cells, and then glucose levels drop in your blood. At the same time, over here, AKT phosphorylates GSK3 beta and turns it off. Now glycogen synthase will get dephosphorylated and it will start making glycogen. So those two things kind of act together in that you're bringing glucose in and then you're allowing the enzyme to make it into glycogen. And now you can store it as glycogen inside the cell. So this is mostly active in like your liver and your skeletal muscle, although all cells will have it as well. Okay, so let me see how my animation is doing here. All right, so my animation, because it's in Flash, I can't really play it normally. It kind of plays through the program that I used to make it, except that it goes on a loop and I can't control it. So I'm going to wait till it comes around to the beginning and then I will share it with you. Here it comes again. I feel like Arlo Guthrie singing, you can eat anything you want at Alice's restaurant. Here it comes around. Best thing about this class is I can tell all my stupid jokes and no one can make fun of me, at least not to my face. Joking all I want. Okay. Okay, here we are. These green things are glucose outside the cell. Insulin binds to the receptor. Receptors dimerize, phosphorylate each other. That brings IRS1 up. It gets phosphorylated. PI3 kinase comes up. Makes PIP3. Notice down here, GSK3 is phosphorylating GS, shutting it off. Okay, now PDK2 phosphorylates AKT, PDK1 phosphorylates AKT. AKT comes over here. Come on, baby. Phosphorylates a protein on this vesicle. This is our GLUT4. Once that's phosphorylated, GLUT4 goes into the membrane, and AKT is shutting off GSK3. Now we've got GLUT4 in the membrane. We're transporting glucose in, and GS is making glycogen out of it. Okay, I'm gonna let this run again without talking. Just watch this bad boy over here, which I didn't talk about before. GSK3 is shutting off GS, and it dephosphorylates and gets phosphorylated again. Constant cycle. I'm gonna add some sound to this too. Okay, phosphorylating that target on the vesicle. Vesicle fuses. transporters in the membrane. Whack. 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 Okay, that's our insulin receptor mechanism. Any questions about that? Unfortunately, I can't show that to you other than the way I just did it. Where are my notes? There, okay. No questions, comments? That was insulin receptor mechanism. Okay, so last but not least is our Wnt mechanism. Wnt. So Wnt is another one of those mechanisms that I talk about here. Like the names of all these things came from Drosophila genetics. So Drosophila development and genetics. And so the names are kind of silly and you just have to memorize them. Like I like things like IRS1, insulin receptor substrate 1, because it says, look, this is a substrate for insulin receptor. Oh, okay, that makes sense. But here we've got things like Wnt, which is wingless, and then we've got frizzled, disheveled, and I guess that's it for weird things. Okay, so anyway, Wnt is a hormone. It's released from one cell onto another cell, locally released hormone, and it's going to again control this destruction complex. So here we've got axin, APC, WTX, and GSK3 beta. So in the absence of Wnt, this is active and is constantly breaking down beta-catenin or phosphorylating it, and then it gets ubiquitinated and then broken down. So here we've got another a complex down here. This is Groucho, which is a repressor. Remember from genetics, the difference between an activator and a repressor. Groucho binds to TCF-LF and blocks it from transcribing. So these genes are repressed. Okay, disheveled is kind of hanging out here. Then we've got LRP and frizzled, which are kind of co-receptors for Wnt, and they're hanging out up here. So Wnt shows up. Wnt binds to frizzled and LRP and makes this complex, which activates or which attracts disheveled. Disheveled then binds to the complex and brings axin up. When you bring axin off the destruction complex, GSK3 beta becomes inactive. Once GSK3 beta is inactive, beta-catenin builds up in the cell really fast, and then it displaces Groucho and activates transcription with TCF-LF. Okay, so basically in the absence of Wnt, our destruction complex is breaking down beta-catenin. In the presence of Wnt, it binds to frizzled, that brings disheveled up and LRP, and that brings axin off the destruction complex, and GSK3 beta shuts off. And that allows beta-catenin to build up in the cell. Okay. Okay. Preguntas, estudiantes. Questions, ask me. No one wants to say a damn thing. No one's saying a damn thing. Question?
I have a question.
Yep. Claire.
I have a question about the insulin receptor mechanism. What is that protein? What was that little receptor at the bottom? The GS beta, I think.
GSK3 beta?
Yeah, that's it.
Okay, that's kind of central to a lot of these mechanisms. Let me pull that up. Nope, that's not it. Sorry. So that's important because it's central to these all three of these mechanisms we just talked about. So we'll start back here with PI3 kinase. This is called the destruction complex, APC, axin WTX, and glycogen synthase kinase 3 beta, okay? So this is going to be phosphorylating either beta-catenin or glycogen synthase, depending on the pathway we're talking about. So this is the PI3 kinase pathway. AKT gets activated and blocks it. This is the insulin receptor pathway. PI3 kinase gets activated a different way, activates AKT, and that phosphorylates GSK3 beta. And then here's the Wnt complex. When Wnt is around, it brings axin off, and that stops GSK3 beta from working. Okay, so that's the same enzyme in all three pathways. It's just regulated or does different things, sort of. Okay, Claire? Okay,
Thank you.
sure. Any other questions? Should I call on one of you to explain it to me? Lake says no. All right. All right, then, a little early, but I think I'm going to call it a day because that's about all I got for you today. So I will see you again on Thursday.
Can you
I got a
go?
quick question, Slash. I don't know if you can answer real quick.
What's that? Go ahead. Did someone just ask me a question? Brian? He's gone. Oh, well.
I have a
I'm
question.
definitely here. I'm definitely here. I don't know if you can hear me, but I was asking what was the last thing you covered on.
Last thing I covered when? Brian, unmute yourself and ask again, please.
What the hell? I'm sorry. It just keeps going. It kept muting itself. But I was asking what was the last thing you covered, because I was trying to keep up on the notes, but I lost where you were. So I was trying to make sure I get all the notes. So I just wanted to make sure.
Okay.
What was the last thing?
The Wnt pathway was the last thing I covered.
Okay. Went pathway. So that was all right. So you finished the notes. All right. That's good.
Okay.
Cool.
And Lake, you got a question?
Yeah, I was wondering if you could just go over the JAK-STAT pathway really quickly again.
Absolutely. So the JAK-STAT pathway is it's almost like it's very similar to the TGF-beta pathway. Basically, you've got your receptor, and the receptor does not have a kinase domain, but when the hormone's around, the two receptors bind to each other, and that brings these kinase domains in. And they transphosphorylate each other, and that activates the kinase even more. And then they phosphorylate the receptor itself. And this then produces an SH2 domain. And that SH2 domain will attract the STATs. So these are the transcription factors. In this condition, they're inactive. They get pulled up by their SH2 binding domain to the receptor, bind to the receptor, then they get phosphorylated. And this is the thing I think is really neat. Now you've got a protein with an SH2 domain and an SH2 binding domain. And so they link up to each other, and that becomes a transcription factor. Okay, does that make sense?
Yeah, thank you.
Okay, good. All right, then. I'm going to let you all go, and I will see you on Thursday.
See you Thursday.